Hello everyone. Good evening. So our today's topic is exercise of biomolecules. We have completed up to question 10. Now today we will do question number 11 onwards. Okay. So first question is means I mean to say 11th question is, what are essential and non-essential amino acids? Actually they are alpha amino acids, right? What are essential and non-essential amino acids? Give two examples of each. This is also very important question. So you know, there are different types of alpha amino acids. Amino acid means it is talking about alpha amino acids. You know, alpha amino acids which are needed for health and growth of human beings but are not synthesized by human body, they are called essential amino acids. Alpha amino acids which are not synthesized by human body. So such amino acids means alpha amino acids, they are needed for health, they are needed for health as well as growth of human beings, but they are needed, but they are not synthesized by human body, means they are obtained from the diet, they are obtained from the diet. So such alpha amino acids are called essential alpha amino acids, right? We have to give example. So can you give some examples of essential amino acids? So say first two, glycine and alanine, they are non-essential alpha amino acids, they are synthesized by our body. Okay, so those alpha amino acids again which are needed for health and growth of human beings but are synthesized by the human body. If they are synthesized by the human body, they are called non-essential alpha amino acids. Okay, and essential alpha amino acids examples are, yes, example is leucine, valine, very good. So keep in mind these two. Non-essential, first two, glycine and alanine, that's it. Okay, simple, which are synthesized by our body, they are non-essential because they are not required from outside, from diet, because they are synthesized by our body. Okay, but non-essential means which are not synthesized by our body. Okay, okay, now next, define. Today I have written the questions. Now what are, sorry, define the following as related to proteins. First is peptide linkage. Yes, peptide linkage, as the name suggests. What do you mean by peptide linkage? You know, proteins, how the proteins are formed. Proteins are formed by condensation reaction between alpha amino acids, and proteins are actually condensation polymers of alpha amino acids, that you know, in which same or different alpha amino acids are connected by CONH bond, and that CONH bond is called peptide linkage or peptide bond. For example, let us take an example. We have glycine, simplest alpha amino acid, that is glycine. NHCH2 C double bond O OH, right? This is glycine. Now glycine, when it is treated with alanine, so OH group of carboxylic and H of NH2. So water is removed. That's why it is called condensation reaction. Means addition of two molecules, two monomers with the elimination of a small molecule. That is called condensation reaction, right? So it become NH2, CH2, C double bond O, NH, CH, CH3 and COOH. So this linkage, is called peptide bond. We call it as peptide bond or peptide linkage. Okay, and this is glycyl alanine, or simply we can write it as GLY ALA. Okay, so this CONH bond is called peptide bond. Okay, CONH bond is called peptide bond. Okay, got it? ठीक है बेटा? Now see this, the this is dipeptide, so tripeptide, tetrapeptide. So chemically a peptide bond is a actually amide linkage. Say CONH2 is amide, so it is actually amide linkage, okay, between COH group of one alpha amino acid and NH2 group of other alpha amino acid by the loss of a molecule of water. Okay, so that is called peptide linkage. Now primary structure. So primary structure means primary structure. So you know proteins जो है that contains one or more polypeptide chains, and each polypeptide chain is made up of large number of alpha amino acids that you all know. Okay, so they are linked by, there is a specific sequence. So primary structure represent the specific sequence of alpha amino acids. Okay, in a specific sequence in which the various alpha amino acids are present in a protein. Okay, and they are linked to one another by peptide linkage. Okay, so that specific sequence in which the various alpha amino acids are present in a protein, okay, that is called primary structure of proteins. Okay, and any change in the sequence of alpha amino acids that create different protein. Okay, means sequence जो है of alpha amino acids that decide the protein. If there is a change in sequence, we get different protein. Okay, so sequence of alpha amino acids that create different proteins. So what is primary structure actually? It tells us about the sequence in which various alpha amino acids are present in a protein. and these alpha amino acids are linked by peptide linkage. Okay. Next, specific sequence. Very good. Now, third part of this question is, what is the third part? Denaturation. So I think denaturation you know all. What do you mean by denaturation of protein? Yes, beta. When there is a change in temperature or pH. Change in temperature or change in pH. Then what happened? Hydrogen bonds are broken. Okay. So due to cleavage of these hydrogen bonds, what happened? Unfolding of proteins. Like globules unfold, and that coiled structure is uncoiled. Okay. And in this way, what happened? Secondary and tertiary structures जो है, they are destroyed, because each protein has a unique three-dimensional structure and has a specific biological activity. Okay. Each protein has a unique 3D structure, and because of that 3D structure, it has unique or specific biological activity. Okay. Unique 3D structure and specific biological activity, right? So when there is a change in temperature or change in pH, then what happened? Hydrogen bonds are broken. Okay. So then that protein, there is unfolding of protein molecules, or we can say coiled structures are uncoiled. Means globular proteins are converted into fibrous proteins, and there is a loss of biological activity. There is loss of biological activity, and that is called denaturation of protein. But keep in mind, during denaturation, secondary and tertiary structures are destroyed, but primary structure means sequence of alpha amino acids, that remains intact. Okay. That means peptide linkage will not break. Okay. So as a result of denaturation, what happened? Globular proteins are converted into fibrous proteins, and globular proteins, they are soluble in water. But fibrous proteins, they are insoluble in water. Okay. Or we can say this denaturation also leads to coagulation. Okay. So coagulated proteins are actually denatured proteins. Any coagulated protein is denatured protein. Most common example is coagulation of albumin present in white of an egg. When egg is boiled, what happened? Soluble globular protein present in it is denatured and is converted into insoluble fibrous proteins. Another simple example is curdling of milk. So boiling of an egg and curdling of milk, these are example of denaturation of proteins. right? Yes, बेटा any doubt up to this? किसी को कोई डाउट है तो पूछ लो. बेटा थियरी मीन्स इतनी थियरी लिखनी है जितनी मैं बोल रहा हूं, है ना? ओके, क्वेश्चन नंबर 13. What are the common types of secondary structure of proteins? Yes, tell me. Common structures of, yes beta, what are the common types of secondary structure of proteins? So secondary structure is what? Secondary structure is, conformations. किसके? Which the polypeptide chains assume when conformations which the polypeptide chains assume, yes, as a result of hydrogen bonding, right? So that is called secondary structure of proteins. So what are common types? What are common types of secondary structure? You can answer. Coagulation, beta. See, egg. In egg, there is a liquid, but when it is boiled, it turns into hard mass. Okay, so that is actually coagulation. Soluble protein is converted into insoluble proteins. That means it is coagulated. Okay. So what are common types of secondary structure? Very good, Ashish. Alpha helix. Common structure of or common types of secondary structures are alpha helix and beta plated sheet structure. Ashish, good. Okay. Now, question number 14. What type of bonding helps in stabilizing alpha helix structure? Yes, alpha helix structure. It is like this, alpha helix structure. So there is a single polypeptide chain which is folded back, and there is a hydrogen bonding between CO and NH. CO and NH. So alpha helix structure is of protein is stabilized by, beta, which hydrogen bonding? Intra or inter? Intramolecular hydrogen bonding, beta. Say, you will write intramolecular hydrogen bonding between CO of one amino acid residue and the NH of the fourth amino acid residue. That you should keep in mind. In the chain, it is intra. Very good. And keep in mind, CO of one amino acid residue and NH of the fourth amino acid residue. Okay, that is called that is the reason of stabilization of alpha helix structure. Okay. Yes, now question P. Question 15. Difference between globular and fibrous protein. Go on writing it. Globular proteins and fibrous proteins. Okay. Yes, write the differences. Write the differences between globular and fibrous. Yes. So in globular proteins, what is the shape? Globular proteins have spheroidal shape, right? Due to folding of the polypeptide chain. Okay, in which polar group is outward and hydrophilic group is outside and lipophilic group is inside. Okay, and here fibrous proteins, they are thread-like molecules. They are thread-like molecules. Means polypeptide chains of fibrous proteins contains thread-like molecules which tend to lie side by side to form fibers. That's why they are called fibrous proteins. Very good. This is the first difference, that globular proteins are spheroidal shape and fibrous proteins are thread-like. Okay. Second, globular proteins are soluble in water, but fibrous proteins are insoluble in water, because in fibrous proteins polar groups are bonded together, but in globular proteins polar groups are outside towards water. Okay. Now, what is the third difference? Globular proteins are more stable or less stable? Obviously, globular proteins are more sensitive to small change in temperature or pH, are sensitive to small changes in temperature and pH. Okay, therefore they undergo denaturation on heating. Okay, or even in the treatment with acids or bases. But fibrous proteins are somewhat stable to moderate changes in temperature. And pH. Okay. Yes. Any other difference? Say globular proteins, they possess biological activity. But fibrous proteins do not have biological activity. Okay. For example, invertase, even hormones, okay, hemoglobin, They are all globular proteins. So they are biologically active, but keratin in skin, hair, salts, even myosin in muscles, they don't have any biological activity. For example, keratin and myosin. Keratin in hair, skin, myosin in muscles, hemoglobin, even maltase, invertase, they are all globular proteins, right? Now, question number 16. How do you explain the amphoteric behavior of amino acids? We have discussed it in length, but once again I am telling you, you know alpha amino acid? Alpha amino acid means in which NH2 group is at the alpha carbon. Carbon next to COH group is called alpha carbon. And you know amino acid contains acidic and basic group both. Okay, in the same molecule there is acidic group as well as basic group. So in aqueous solution they neutralize each other. So what happened? The COH group that loses H plus ion and NH2 group that accept H plus ion. As a result, it is like this. So it exists as dipolar ion or we can say zwitterion structure. Okay, that is called zwitterion structure. So in zwitterionic form, alpha amino acid show amphoteric behavior because it can react with acid as well as base. Okay, like see, this is the zwitterion structure of alpha amino acid. In acidic medium, what happened? In acidic medium, you know, it exists as cation. That means it can react with H plus ion, or in basic medium it exists as anion, right? So because of the presence of this zwitterion, it can react with acid as well as base. Okay, so NH3 plus act as the acid and CO negative act as base in the zwitterion. So it can react with acid as well as base. That's why it is an amphoteric molecule. Means it shows amphoteric behavior of amino acid because it has acidic group as well as basic group. Okay. Next, what are enzymes? First of all, enzymes are biological catalysts. Enzymes are actually catalysts, but they are biological catalysts, right? They are biological catalysts. And you know, each biological system requires a different enzyme. You know that each biological system requires a different enzyme. Okay, enzymes are, first of all, enzymes are very efficient. Enzymes are very good beta, efficient and are specific at the same time in their action. For example, you know, um, let's take one example like invertase. So invertase is used in the hydrolysis of cane sugar. It cannot be used to hydrolyze maltose. To hydrolyze maltose, we need maltase. Okay, so we cannot interchange, right? And you know, they are required only in small quantity. They are required only in small quantities. They are not required in excess, and they work at only optimum temperature and optimum pH. You know what do you mean by optimum temperature and optimum pH? Like if we draw the diagram. So this is called optimum temperature. Means rate is slow below this temperature and above this temperature. Rate is maximum, rate of the reaction is maximum at that particular temperature. So that is called optimum temperature. Same rate versus pH. Again the same thing. Rate is maximum at a particular pH. Below that pH, above that pH, rate is slow. So that is called optimum temperature and optimum pressure. And chemically, enzymes are actually globular proteins. This is the main, because you know, globular proteins have biological activity. Okay, so chemically they are globular proteins, and they are also associated with some non-protein component that is called cofactor for their activity. We require, they are also associated with a non-protein part. Actually they are proteins. Okay, but in addition to proteins, they are also associated with a non-protein part that is called cofactor for their activity. And cofactors are of two types. One is inorganic ions. I am talking about non-protein part that is attached to, yes, enzyme. Okay. Wow, beta, wow, very good here. So cofactors like inorganic ions. Yes, tell me any inorganic ions like Mg2 plus, Zn2 plus, even copper 2 plus, CO2 plus, Fe2 plus, they are inorganic ions required for the activity. Or organic molecules. Those organic molecules are called coenzymes or prosthetic groups. Organic molecules which act as a cofactor are of two types, either coenzymes or prosthetic groups. So coenzymes are actually vitamins. Okay, coenzymes are vitamins. Okay, they are loosely held to proteins and can be easily separated by dialysis. And prosthetic groups, they are derived from vitamins like biotin, but they are tightly held to the protein molecules by covalent bonds. Say, coenzymes are loosely held, okay, and they are easily separated by means of dialysis, okay, but prosthetic groups are, they are tightly held to the protein. By covalent bonds, okay, like biotin, okay. And give example of coenzyme. Any example? Like riboflavin, okay. Oh, 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 okay, beta. Sure. I will keep this in mind. Khalil, can you send me this on WhatsApp? This is my mobile number. You can send this on WhatsApp so that I will try to make a video on it. Okay, Khalil? Yes, so this is question number 17. Okay. So prosthetic groups, they can be separated only by careful hydrolysis. Say this coenzyme can be separated by simply dialysis, okay, but this can be separated only by careful hydrolysis, and this can be separated by dialysis, okay. So this is the question number 17. Now question number 18. What is the effect of denaturation on the structure of proteins? We have just discussed it. During denaturation, what happened? During denaturation, secondary and tertiary structure of proteins, they are destroyed, okay, but primary structure remains intact, okay. And as a result of denaturation, what happened? Globular proteins, they are converted into fibrous proteins. And you know, secondary and tertiary structures of protein decide the biological activity. So their biological activity is lost due to denaturation. For example, boiled egg that contains coagulated protein cannot be hatched to produce chickens. See, egg, when it is not boiled, that can hatch to produce chickens, okay, but if egg is boiled, can we produce chicken? Boiled egg cannot be hatched to produce, boiled egg cannot be hatched. To produce chickens, okay. That's it. Now question number 19. How are vitamins classified? This year this question will not come, okay, but it is important for your competitive exams, right? So let us revise it for the competitive exams, okay? How are vitamins classified? Vitamins are classified into two groups depending upon their solubility in water or fat. There are two types of vitamins. First, water soluble vitamins. Water soluble vitamins and fat soluble vitamins. Okay, we have two types of vitamins. So question is, how are vitamins classified? So you should write vitamins are classified into two groups depending upon their solubility in water or fat. And water soluble vitamins, they include most common vitamin B complex. Vitamin B complex, for example, B1, B2, B3, likewise, okay, and vitamin C. And fat soluble vitamins, they include, yes, they include vitamins A, D, E and even K, and they are stored in, they are stored in liver, okay, and adipose. That is fat storing tissues. Okay. However, why biotin? You know biotin? What is biotin? That is vitamin H. It is neither soluble in water nor in fat. Okay. Vitamin H is neither soluble in water nor in fat. Keep in mind. Okay. And vitamin K, it is responsible for coagulation of blood. That you should keep in mind, right? Very good, Ashish, good. Now question number 20. Why are vitamins A and vitamin C essential to us? Yes, tell me why they are essential to us and give their important sources. Yes, tell me why vitamin A and vitamin C are essential to us. Yes, beta. Simple, because their deficiency can cause some disease. For example, vitamin A. If a person has deficiency of vitamin A, then its deficiency causes, it is essential to us because, yes, its deficiency causes, can you tell me? Very good. It causes night blindness. Okay, I think you have done this chapter before, because xerothalmia, what is xerothalmia? Can you tell me? What is xerothalmia? It is, yes, it is hardening of cornea of eye. Okay, it is caused due to the deficiency of vitamin A, and vitamin A is in butter, milk, even carrot. You have written all. Uh, I'm blessed to have students like you. Okay, and vitamin C. In vitamin C, it is again essential to us because its deficiency causes this. Everybody knows, I think, because it is in class 9th and 10th as well. Scurvy means bleeding gums or pyuria. Right? That is loosening and bleeding of teeth. And it's obviously citrus fruits, all citrus fruits like amla. Okay, good, lemon, very good. So they are sources of vitamin C. Okay, all the citrus fruits. Good, beta. Now question number 21. What are nucleic acids? Mention their two important functions. Nucleic acids. What are nucleic acids? As the name suggests, see, carbohydrates, proteins, nucleic acid, they are all biomolecules. And these are the biomolecules which are found in nuclei of all living cells. Okay, in the form of nucleoproteins or, yes, nucleoproteins or chromosomes. Okay, and this chromosome, these are the proteins containing nucleic acids as the prosthetic group. Okay, nucleic acids are of two types. Yes, DNA and RNA. DNA means deoxynucleic acid, deoxyribonucleic acid, and RNA means ribonucleic acids. And what are their functions? Yes, DNA is responsible for the heredity effect. Very good. From one generation to another. And this is due to the property of replication. DNA has property of replication because it is double stranded, you know. Okay, replication during cell division, as a result, two identical DNA strands are transferred to daughter cells. That we have done all. Okay, and DNA and RNA, they are responsible for the synthesis of proteins, all the proteins, because actually synthesis of proteins is carried out by RNA, but its message is coded in DNA. Okay. So proteins are synthesized by various RNA molecules, ribosomal RNA, messenger RNA, transmission RNA. Okay. But message for the synthesis is coded in the DNA molecules. That's it. These are the two functions. Okay. Now, question number 22. What are the difference between a nucleoside and a nucleotide? So, what do you mean by nucleoside? In nucleoside, it is sugar plus base, and nucleotide is sugar plus base plus phosphoric acid, or we can say phosphate. So that is nucleotide. Simply you can write it like this is sugar. Okay. At second position, it is OH or HH, that all of you know. Here at 1 dash there is base, at 5 dash, okay. So nucleoside means sugar plus base. So nucleoside is formed when one position of the pyrimidine or 9 position of the purine, say base, one position of pyrimidine and 9 position of purine. Okay, that is attached to C1 of sugar, ribose or deoxyribose by beta linkage. This is beta linkage. Okay, and nucleoside is sugar base only. And in nucleotide, see, this is H, OH, 1 dash, 2 dash, 3 dash and 4 dash. H, OH, 1 dash, there is a base, 2 dash, 3 dash, 4 dash, and at 5 dash there is phosphate group. Okay, sugar, this is sugar, base, and this is phosphate. So nucleotide consists of all the 3 basic components of nucleic acids: a phosphoric acid group, a pentose sugar, a nitrogenous base. See, there is pentose sugar, okay, a base and phosphoric acid group. Okay, and they are obtained by the esterification of C5-OH group and pentose sugar by phosphoric acid. Done? Sugar plus base plus phosphoric acid. Very good, Charitre. Okay. Now question number 23. The 2 strands in DNA are not identical but are complementary. Can you explain? You know, 2 strands of DNA, they are, yes, 2 strands of DNA, they are held together by hydrogen bonds. They are held together by hydrogen bonds between purine bases of one strand and pyrimidine bases of other strand. Purine base of one strand and pyrimidine base of other, and vice versa. And because of different sizes and geometry of the bases, the only possible pairing in DNA is G and C. If there is a G and C, there is a 3 hydrogen bonds, and between A and T there are 2 hydrogen bonds. Okay, like if there is T on the other side, there should be A. If there is side C on the other side, there should be G. And due to this base pairing principle, sequence of bases in one strand automatically fixes the sequence of bases in the other strand. Like if sequence is G A on one side, G A T C, so on the other strand, this is first strand. Okay, sorry, strand 1. So on the strand 2, what is the sequence? Corresponding to D, there is a C, A, there is T, T, there is A, C, there is G. So this is the sequence on the other strand. Means sequence in the one strand automatically fixes the sequence of bases in the other strand. So that's why the two strands are complementary and not identical. If it is 3 dash, it should be 5 dash. Okay. So if it is 5 dash, it should be 3 dash. That's it. Got it? So they are complementary to each other because of the base pairing principle. Okay, because G always connected with C through 3 hydrogen bonds, and A is connected to T. A of one strand is connected to only T of the other strand, right? Because of different sizes and geometry of the bases, right? That's why they are complementary to each other and not identical. Question number 24. Write the important structural and functional differences between DNA and RNA. DNA and RNA. So in DNA, can you tell me what sugar present in DNA is? 2-deoxy D-negative ribose. And in RNA, the sugar present is D-negative ribose. And DNA contains, yes, DNA has double-stranded alpha-helix structure. DNA has double-stranded alpha-helix structure. Okay, and RNA has only single-stranded alpha-helix structure, right? In DNA, bases present are at Chandigarh, but in RNA, bases present are ao Chandigarh. Adenine, thymine, cytosine, guanine. Adenine, adenosine, uracil, cytosine and guanine. Okay. DNA molecules are large and RNA molecules are much smaller. Okay. their molecular mass is 6 into 10 power 6 to 16 into 10 power 6. But in RNA, it is 20,000 to 40,000 only. Okay. And now function, this is the structural differences. Okay. Now functional differences. You know, what is the functional difference between DNA and RNA? In DNA, they have unique property of replication. DNA has unique property of replication, but RNA does not replicate. Okay. Second functional difference is DNA that controls the transmission of heredity effect. It control the transmission of hereditary effect. Okay. From one generation to the other. But RNA, it controls the synthesis of protein. Okay. Okay, beta. Thank you for your patience. Last question of the day. Okay. Oh wow, Charitra, you are great, beta. Question number 25, last question of this chapter. What are the different types of RNA found in the cell? So there are, yes, 3 types of RNA, right? Those are ribosomal RNA, that is also called RNA. Okay. And second is messenger RNA, that is mRNA, and last is transfer RNA. Very good. That is tRNA. Okay, boss. Bye bye, beta. This is all about this chapter, that is biomolecules. Okay. So bye bye to all. God bless you. Thank you so much. Good night.